Have you ever wondered what's in contaminated water? Well, today we're going to find out what's in the water by Brianna Kurtz. What is the purpose of the experiment? The purpose of the experiment was to identify unknown compounds detected in a drinking sample supplied by contaminated surface water using infrared and nuclear magnetic resonance spectroscopy. Each water sample had different kinds of pollutants in it that contaminated the water. What is IR spectroscopy? IR spectroscopy is when an infrared radiation source is shown through a solution and the photon absorption rates are taken on the other side. This data is then put into a graph. The graph shows the different spikes on it. Each spike is representative of a functional group. That's what this arrow is pointing to. As you go farther right on the graph, this becomes known as the fingerprint region. This is more inconclusive data and it becomes harder to tell which spike is which functional group. For this case, for our experiment, the Nicolette IS5 FT IR spectrometer is what was used. What is NMR spectroscopy? NMR spectroscopy is when a sample is introduced to a magnetic field and light is shown through and then detected on the other side. It then produces the graph that is shown below in figure 2. As you can see, there are two spikes. Each spike is representative of hydrogen nuclei reacting with a magnetic frequency and the proton alignment. The number of peaks, which in this case would be 2, gives you the types of H. And the ratio of peaks, which would be 1 to 1 in this case, gives you the number of H nuclei in each peak, or the type of H. And for our experiment, the NMR spectroscopy was collected at 60 MHz on an EFT 60 spectrometer. So what were the results of our experiment? How were the unknowns found? This is example unknown 31420. First, you can use NMR. This is the data for that unknown. You have 2 peaks, which you can see evident in the figure 2 here. The ratio of areas, which is 1 to 1. The number of peaks and the ratios of areas when compared to the data graph gives you methyl acetate or 1,4-dichlorobutane. The second way of analyzing the solution was through IR spectroscopy. The sample data is shown above. It shows that a CH singly bonded carbon was found. This is evident through the yellow highlighted part between the 3010 inverse centimeters and the 2800 inverse centimeters. The CCl is also evident in between the 800 inverse centimeters and the 600 inverse centimeters. This is highlighted in green on the graph. Based on these checked boxes, it was either 1,2-dichloroethane or 1,4-dichlorobutane. This chemical yielded that based on the NMR and IR, only 1,4-dichlorobutane could have been the possible substance. This diagram to the right here is the chemical structure of dichlorobutane. In this chemical structure, you can see the evidence of the CCl bond, and this is good. This means that we did our jobs correctly. This is a table of the nuclear magnetic resonance spectroscopy. This is what each value was used to compare back to, and this was also used for all of the other unknowns. This is table 2, infrared spectroscopy with all the values and the checked values. This is a table that was used to refer back to, just like the one on the slide before, the NMR. This was also used for all the other knowns as well. So this is table 3, IR spectra of unknown compounds. It lists the functional groups for each sample. For sample 2, we had a CH benzene ring, a CH singly bonded carbon. For a CH3, we had a CH singly bonded carbon and a C double bond O, and we already reviewed sample 1, so that's why I wasn't going through it again. For table 4, NMR spectra of unknown compounds. So for number 2, the number of peaks was 2, with the ratio of areas to the peaks was 3 to 2. And then for sample 3, the number of peaks was 3, with a ratio of areas of 3 to 2 to 1. This is our second sample, unknown 60465. As you can see in figure 3, the blue highlighted part is the CH benzene ring between 3,150 inverse centimeters and 3,000 centimeters. The The CH is the yellow highlighted part with a singly bonded carbon of 3010 inverse centimeters to 2800 inverse centimeters. And then for figure 4 of the NMR, it shows 2 peaks with the ratios of areas of 3 to 2. With this data, it narrowed down the choices to paraxylene. Again, this is the chemical compound shown below of paraxylene, which further solidifies that we did everything correctly because it shows the CH groups and the benzene ring. This is the third unknown 94086. For the IR spectra, it shows that in the yellow highlighted area is the CH singly bonded carbon between 3,1000 inverse centimeters and 2,800 inverse centimeters, and then the C double bond O is 1,750 inverse centimeters and 1,650 inverse centimeters. It's highlighted in the light purple. It's also a very strong peak. Figure 6 is the NMR spectra. You can see that there are 3 peaks, very evident. The one is obviously larger than the other. If you look closely, one has 1 singular peak, the second has 2, and then the third has about 3 stronger ones. So this would mean that the ratio of areas is 3 to 2 to 1. With this data, we narrowed it down to the chemical being ethyl formate, and this is the chemical structure below. All of our data is evident in it, so we did it right. For each of these possible contaminants found, there could be health risks. For sample 31420, 1,4-dichlorobutane, there could be burning of the skin and eyes, irritation to the lungs, dry and cracked skin, redness, or water blisters. For ethyl formate, there could be irritation of the nose and throat, coughing, shortness of breath, and a possible buildup of fluid in the lungs. It can also affect your nervous system, which is not good. There could also, for sample 60465, which is paraxylene, it could be a skin irritant, coughing, wheezing, headaches, nausea, vomiting, and dizziness. It could also be a reproductive hazard. In the grand scheme of events, I would say that paraxylene or ethyl formate is the most destructive to humans. Each of these compounds can also cause effects on the environment. 1,4-dichlorobutane can be harmful to aquatic animals. Ethyl formate is very low toxic and low risk, so it really wouldn't be that dangerous. Paraxylene leaks into soil, surface, or groundwater, evaporates, gets broken down. So eventually, it just evaporates into the air, so there's not really a risk with that one. So in conclusion, IR and NMR spectroscopy are valuable tools to use when identifying contaminants in water. Our experiment yielded that 1,4-dichlorobutane, paraxylene, and ethyl formate were all the most likely compounds. This was made possible through highlighting the functional groups, the number of peaks, and the ratio of peaks found in the NMR and IR spectroscopy. Through this information, we were also able to do research on the health risk and environmental risks. I would say that 1,4-dichlorobutane is the most overall risk. However, paraxylene does pose a huge risk as it does affect the reproductive system. Then this is a page of my references. Thank you for watching.